Hi everyone. Let's learn a little bit about chromosomes today. I'm pretty much going to speak on the basics. So we're going to learn about what are chromosomes, describe chromatin packaging, explain what are autosomes and sex chromosomes, what is meant by diploid and haploid. We are going to look at the classification of chromosomes, what's an ideogram, and samples used for chromosomal studies. We are going to look at the methods of chromosomal study, the nomenclature of chromosomes, and a brief introduction on chromosomal aberration. So, what basically are chromosomes? Chromosomes are nothing but DNA material, as we all know, right? But then why don't we directly call it DNA? Why do we call it chromosomes? Let's look at it, where it all starts. So, it's like there are small nucleotides, right? The nucleotides are basically in the DNA A, G, C, and T. We all know that A pairs with T and G always base pairs with C. Now, these nucleotides, the nitrogen bases which make up the nucleotides, now make up a double-stranded DNA. A segment of this double-stranded DNA, like say just this segment, starting from this end, this can be one gene. The next segment can be another gene. Okay, this next segment can be another gene. Now, this DNA strand coils or folds further, right? And they form, because of this folding, the chromosome is being formed. Okay, the chromosome is being formed. And this chromosome is inside the cell. And in human body, we have millions of cells, of course. Now, however, the folding of the DNA into a chromosome is not just a single step. In the upcoming slides. But what does the word chromosome means? Some, as it's derived from the word soma, means body. Chrome means colored. Okay, so it's basically described as a colored body, which was seen in the cell nucleus. In total, we have 46 chromosomes, out of which 44 are autosomes and 2 are sex chromosomes. In females, you have 2 XX chromosomes, and in males, we have 1 X and 1 Y chromosome. Right? This is the scientist Henrik Wilhelm Gottfried von Hertz who discovered, or who named the chromosomes as chromosomes. In the previous slide also. Like, these are the histones around which the DNA will coil around. This histone protein code that we have here is actually, though it looks like one unit, it's not. It's a octamer. Okay, what it is called? It's called a octamer. Meaning what? It has, octa means 8, meaning it has 8 subunits. Okay, there's 8 subunits. Now, what are the 8 subunits made up of? It's basically made up of, you can say, 4 types of proteins. There are histone. One is H, okay, 2A, 2B, 3, and 4. Okay, these are the 4 proteins, histone proteins, that make up the histone octamer, and these are present in duplicates. That's why 4 proteins, 8 subunits, and that's why it's called an octamer. Now, approximately around 140, I think to be precise, it is 147. 147 base pairs of the DNA coils around this histone and it makes 2 turns around the histone. Okay, and before it moves on to the next histone, it leaves out a spacer DNA region. To this spacer DNA region is attached the H1. You can see the H1 histone protein is there, and of course there are non-histone proteins also around. This spacer region is around 60 base pairs approximately. It's around 20 to 60 base pairs. It's mentioned at the third point of the slide. Now, so this unit where it's around, the DNA coils around the histone, this unit is called as a nucleosome, which is literally the basic structural unit in the chromatin molecule. Okay, now when we have repeated nucleosomes like this with spacers in between, it definitely gives the appearance of a beads on string appearance, right? It's like beads hanging on a string. So this appearance is also called as beads on a string appearance of the nucleosome fiber. This nucleosome fiber further coils up, it just coils up, it piles up, but in concentric way. You can see it's rather like this, something like this. It coils up on top of one another. Okay, now when it coils up, it gives us a structure. It gives us a structure which is called the solenoid. This solenoid is nothing but a cylindrical structure like thing. Okay, it forms a cylindrical structure, and that's why it was named as solenoid. This solenoid further forms loops. Okay, this further forms loops and it goes inside. It goes inside where? It goes inside the cell, forming the loops, right? And when it's diffused like this, it's not condensed, the DNA. That is seen when? In the interface of the cell cycle. Okay, where we see euchromatin, you can see. Now, like I told you, the solenoid is cylindrical, and the word solenoid itself was derived from the Greek word solenoids, okay, which means pipe-shaped. Is nothing but the secondary helical structure. Okay, like I told you, the slide mentions that solenoids themselves are packed into loops or domains attached at intervals of approximately 1000 base pairs to form a protein scaffold within the nucleus. Okay, and it goes inside the nucleus. The H1 histone proteins that I told you about, you can see here, is seen here in the spacer region. Okay. Now, coming to the concept of euchromatin and heterochromatin. What is euchromatin? Euchromatin is nothing but the decondensed chromatin. Heterochromatin is nothing but the condensed chromatin. Okay. Euchromatin is the coding DNA. Heterochromatin is the non-coding DNA. And in interphase, the DNA is most decondensed. Chromosomes are maximally condensed in the metaphase. Now, while euchromatin, the decondensed form, is the coding DNA. Let's take a simple example. You are painting on a piece of fabric. Will you be able to paint when the fabric is open or the fabric is closed? Consider the closed as condensed. Of course, you will be able to paint when it's open. It's decondensed, right? Similarly, when our DNA is decondensed, various transcription factors can attach along the DNA sequences and help in coding the DNA to the RNA. Okay, and that's why euchromatin is also the decondensed. In decondensed chromatin, you can code or you can transcribe the decondensed DNA. Whereas heterochromatin, which is the condensed form, which is the closed form, you can say, like the closed fabric. We cannot paint on a closed fabric, right? Similarly, on a condensed DNA, the proteins cannot code. And that's why you can call that as the non-coding DNA. And usually, euchromatin is seen in interphase. Okay, and in metaphase, which is one of the phases in the cell cycle, the chromosomes are maximally condensed and they align at the center. Okay, they align at the center. This is metaphase. And when the cell N is born, you can say, okay, it directly goes into the interphase, you can say. Right after it's born, it gets into the G1 phase, then it gets into the S phase, synthesis phase, where DNA is being synthesized, and then it's open for transcription, you can say. So it's the interphase it starts with. Now, this metaphase that I talked about is, since the chromosomes are maximally condensed, you can see it very clearly. Okay, the structure of the chromosome, and it is the best phase to study the chromosomes, I would say. And actually, clinically, in metaphase, we do karyotyping. Okay. Other comes from the father. Now, these chromosomes, 1 to 22, are called as autosomes. Okay, both the chromosomes, both the chromosomes that I have here, the chromosome 1, chromosome 1, like both chromosome 1s are alike morphology. Both chromosome 2 have the same morphology. And the XX chromosomes or XY chromosomes in male are called the sex chromosomes or the gonosomes. Of course, the X and X chromosome in female, both the X's have same morphology, but in males, the X chromosome and the Y chromosomes, both the sex chromosomes have different morphology. Of course, one being X, other being Y. Okay. So now what is haploid? Haploid is nothing but it's not in pairs. Okay. It's just 23 chromosomes in a haploid cell. What are the haploid cells in the body or the human body can produce? It's nothing but the gametes. Okay, the ovum and the sperm. The ovum is also haploid. It has 23 chromosomes. And the sperm is also haploid, 23 chromosomes. That is why when a ovum is being fertilized by a sperm, 23 chromosomes from the sperm fuses with an ovum which has 23 chromosomes, and now they make 46 chromosomes. Okay, out of which, of course, 44 are autosomes and 2 are sex chromosomes. So if you are asked, how many autosomes does a gamete have? The answer is 22, not 44 or 22 pairs. Okay, along with how many gonosomes or how many sex chromosomes? Just one sex chromosome. Either it's a X or a Y. Now, just for a thought, do you think ovum can have a Y chromosome? Can ovum have a Y chromosome? No, a ovum will not have a Y chromosome because ovum is produced from whom? The mother. Does the mother have Y chromosome in the body? No, mother does not have Y chromosome in the body. So the ovum can have only a 22 autosomes and one sex chromosome, which is the X. Whereas the father, can he produce sperms with a Y chromosome? Yes, he can produce a 22. Why? Can he produce a gamete, a sperm with an X chromosome? Yes, he can. 22 and an X. Why? Because males have 44. Males have 44 autosomes and X and Y in men, right? In men. So they can produce gametes with a Y as well as an X. However, women can produce only ovum with 22X. Okay, so when a gamete fuses with a 22X ovum, then the zygote will develop into a boy or girl, into a male baby, a boy. But when a 22X is being sperm, then what will happen? A female baby, a girl will be born, right? Making the zygote, of course, diploid. And then from the zygote, everything else becomes diploid, right? Except that in the ovum, in the ovary and in the testis, later on, what will happen? The gametes produced will be haploid. To know how diploid cells become haploid, how a gamete is produced, read up meiosis. Okay, the process of oogenesis and the process of spermatogenesis. Okay, now chromosomes are classified, like I told you, based on the centromere position, based on their size, and based on their banding pattern. Size we understand, big or small or medium, whatever it is. Centromere, I told you, the constricted region in the chromosome. Banding pattern, what is banding pattern? Nothing but when the chromosomes are stained, it takes up it makes up a banding pattern. In certain regions, the stain is taken up more. In certain regions, the stain is taken up lesser. So it forms bands across the chromosomes P arm and Q arm, you can say. And based on that banding pattern, the chromosomes are classified or identified. Now, usually based on the size, if you observe, the chromosome 1 in this picture is the biggest, and chromosome 22 looks like it's the smallest, right? Based on the size, you can say chromosomes are numbered from the largest to the smallest, 1 to 22. It's certain exceptions in between where chromosome 10 and 11 are a little shorter actually than chromosome 12, and chromosome 21 is also a little smaller than 22. In X and Y, X is bigger, Y is smaller. Okay? Based on the centromere position, if the centromeric constriction is right at almost at the center, okay, it's almost at the center, then it is called a metacentric chromosome. Metacentric. Remember, like a mnemonic, M for metacentric, M for middle. It's in the middle almost, where P and Q arms are similar. Okay, and then the centromere is a little away from one of the arms, and the shorter arm, P arm, is of course shorter, and the Q arm is the longer arm. Okay, this is called as submetacentric. If the P arm is extremely short, like satellite chromosomes, chromatin regions, then these kind of chromosomes are called as acrocentric chromosomes, where the Q arm is very long and P arm is very short. Then there is something called as telocentric chromosome, where the P arm does not even exist. The centromeric constriction is just at one end, and the chromosome has only Q arm, you could say, right? This is telocentric. However, this does not exist in humans. Okay, remember, this does not exist in humans. What we have is only acrocentric, submetacentric, and metacentric chromosomes. Okay, now based on the order of the size and the position of the centromere, the chromosomes were classified into various groups. Okay, and the groups are 7 in number from A to G. A, B, C, D, E, F, G. Okay, in this, if you look 1, 2, 3, they look like how? They look like metacentric. Of course, they are metacentric. 1, 2, 3 was classified as group A. Metacentric, large. So large metacentric chromosomes. Okay. 4 and 5, the P arm and Q arm are not equal. So submetacentric, large submetacentric, classified in group B. 6 to 12, these were classified as submetacentric, medium-sized, along with the sex chromosome X. So 6 to 12 along with X is group C. Okay. 13, 14, 15. What kind of chromosomes are they? They are acrocentric. Are they very big? No. Are they very small? No. They are called as medium-sized acrocentric chromosomes with satellites. These are classified as group D. Group E, you can see that there is a mix of metacentric and submetacentric chromosomes. And 16, 17, 18 is classified as group E, okay, and they are called moderately short metacentric, submetacentric chromosomes. 19 and 20 are short metacentric chromosomes, okay. If you see, both P and Q arms look similar and they are short, so called short metacentric. 19, 20, group F. And what is group G? Nothing but the short acrocentric chromosomes, which is 21, 22, and the Y chromosome. Okay. Now, in the human body, Why are these chromosomes important? Why are we learning about chromosomes? Because in future, when you become doctors, you're going to learn about chromosomal disorders, which are nothing but a result of anomalous chromosome, either in the number of chromosomes or in the structure of chromosomes. Okay, and in such cases, we have to do a chromosomal analysis to diagnose. Okay, to look if the structure is normal or if the numbers are normal. Okay, now for chromosomal analysis, what we mainly use is blood or saliva. From blood, can we use RBCs? No, we cannot use RBCs. Why? Because RBCs don't have nucleus, and when there is no nucleus, there is no chromosome, right? So we'll take the white blood cells which are nucleated, okay, and use for chromosomal analysis. From buccal mucosas, saliva, in the saliva, buccal mucosal cells are present. So saliva is collected, or in clinics, usually when they need to collect buccal mucosal cells, they just ask the patient to rinse out a little water from their mouth into a sterile container. So that's how they collect the buccal mucosal cells. Okay, and blood collected is usually peripheral blood from the peripheral veins, the regular blood that you give during blood testing. Okay, it's now, what are these prenatal samples? The word pre means what? Prior. Natal means relating to pregnancy. Relating to the, natal means birth, and prenatal means relating to pregnancy. Okay. Why do I need to collect samples when the mother is pregnant? Let's say if someone has a history of chromosomal disorders in the family, okay, to make sure the current baby that's on the way is healthy, okay, patients would like to do a prenatal testing, okay, so that they don't end up having a second baby with a chromosomal disorder. Or else, say, if the mother's age is higher and there is a suspicion of syndrome like Down syndrome, where there is 3 copies of chromosome 21, okay, you want to know for sure, like, if the baby is affected or not, okay, then we can do a prenatal testing. Okay, of course we can do prenatal testing in many other genetic disorders, but for chromosomal analysis also in the unborn baby, we need to do sometimes prenatal diagnosis where we need prenatal samples. In prenatal diagnosis, we can use either the amniotic fluid or the chorionic villi sample or the cord blood or cell-free DNA. Now, what is amniotic fluid? Actually, if you know, if I'm sure if people have studied and you know that there is something called the placenta, right? On the placenta, the baby is attached. Now, let's say very bad at drawing, okay? This is our baby, okay, attached to the placenta. Now, around this baby, there are layers called as the amnion and chorion, right? And surrounding the baby, there is fluid around all this region. That is nothing but the amniotic fluid. So that amniotic fluid has been collected for prenatal diagnosis. And from the placenta, the chorionic villi of the placenta, we can take a chorionic villi biopsy, which is called as chorionic villi sampling. Okay, all of this can be done transabdominally. Okay, but we can also try to do sometimes the chorionic villi sampling can be probably done transvaginally also, transcervical. Or else we can also collect blood from the cord that is connecting the baby to the placenta. Okay, that is also done transabdominally. Now, what is cell-free DNA? The baby is growing inside the mother's body, right? That means it's getting blood from the mother. So when the baby is growing, when the blood is being circulated from the mother, the baby's blood is also circulating within the mother's blood. Okay. In such a case, what happens? Some of the epithelial remnants from the baby can freely circulate in the mother's blood. Okay. And those remnants, the epithelial layers and everything that is being shed off, those cells will release its DNA when it's broken down, right? And that DNA keeps circulating in the mother's body. And that is called as cell-free DNA, which also can be collected and nowadays is being commonly used for, you can say, in many institutes for detection of trisomies. Okay, especially trisomy 21, 16, and 18, it has been found useful. So that was about sampling. Now, we'll collect the sample, but how will we analyze it? We'll analyze by techniques called as, there is something called as karyotyping, okay, that we'll look at. There is some fluorescent in situ hybridization, chromosomal microarray, and next generation sequencing, okay, with a low coverage can be used for chromosomal analysis. Can be done in the first point in metaphase or prometaphase. Usually, a high resolution karyotype is done in prometaphase. We arrest the cells in prometaphase and collect the chromosomes. Okay, and a normal resolution karyotype is done in chromosomes together. Okay, the structure of all the chromosomes together. Not see all the chromosomes together. Okay, so the limitation of FISH is that it's region specific or site specific. I can look only for a particular site. Okay, it can be in the metaphase, it can be in the interphase also. Doesn't matter, because if that region is present, it will bind. And in fish, I'm not looking for the solid structure like in karyotype, so I need not have to do it in metaphase. I can do it in interphase also of the cell site. We said duplication in XQ2A, meaning what? Chromosome X, Q arm, region 2, band 8 had a duplication. Had the duplication been here, okay, and this corresponds to 27.3. That means what? In chromosome X, in region 2, band 7, subband 3, there is a duplication. And the previous band is Q27.1. If it was here, it would be chromosome X, Q1, region 2, band 7, subband 1. And in between this subband, there is 7.2, of course, Q27.2, right? So each arm is divided into regions, say 1, 2, or whatever. This ISCN is nothing but International System of Cytogenetic Nomenclature. Okay? According to that, if say a region of the chromosome is missing, deleted, it's called a deletion, of course, right? And it's abbreviated as DEL. If it's duplicated, it's written as DUP. Okay? If there is an inversion, it's written as INV. So there are, for various anomalies within the chromosome. The anomalies are nothing but are abbreviated. Okay, and by anomalies I mean deletion or duplication or a break, whatever it is. Okay, so chromosomal abnormalities, like I told you, it can be in the structure of the chromosome or in the number. Usually we should have two chromosomes of chromosome 1, two of chromosome 2, two of chromosome 3, so on until chromosome 22. And we should have, in girls, in female we should have two X chromosomes, and in men we should have one X and one Y. Any aberration from this normal is an abnormality. So if I have an extra chromosome, say let's say in chromosome 18, here we have another copy of 18. Okay, then that means I have 3 copies of 18. This 3 copies of 18 would be what? It's an abnormality. Okay, which is nothing but a broadly classified as aneuploidy, called as trisomy 18. Okay, so trisomy can be in any of the chromosomes. Okay, and it's always abnormal, it's never normal. Structural. What is structural? Say from this chromosome 1, this band is missing. That means what? This band is deleted, right? So it's a structural abnormality, deletion. Okay, or say in chromosome 2, this region is duplicated, this N band, light band is duplicated. So it's a duplication. Inversion. What is inversion? So deletion here is nothing but we have this chromosome, a portion of it getting lost. Okay, this is a deletion. If a single nucleotide is not lost, small nucleotide is lost, which is not visible in the structure, it can be called a microdeletion. Okay, those are usually point mutation, single. Okay, macrodeletion, which can be seen in the chromosome, a big portion, chunk of the chromosome itself is lost. That is a macrodeletion. This is nothing but a representative diagram of a chromosome 22 where in the region 1, 1.2 is being lost, but you don't really need to remember this, but remember 22, in chromosome 22, a deletion in the Q arm at region 1, band 1, subband 2, 1.2 leads to a syndrome called as the 22q deletion syndrome or the DGeorge syndrome. Okay. That region is also prone for duplication sometimes. Okay. And in duplication, what happens? Say this region is there in the normal chromosome. This becomes double. Okay. Or if there is a sequence, the sequence is a one orange region. We have another orange region within the gene duplication. Okay. Inversion. What is inversion? Look at this chromosome. See this dark orange bands? They are above. Light orange is below. Now if there is a break here and there is a break here, and this reverses itself and inserts itself into the chromosome. So now it's inverted. So it's called the inversion. How would I write? Say we have the number of chromosomes is correct, but this is an error. We have to write it, right? So that in such a case, we'll write how? 46, total number of chromosomes, XY, that means it's a men, inversion 9. What does this mean? That means there is an inversion in chromosome 9. Where in chromosome 9? In the region involving the P arm at region 1, band 1, to Q arm 1, 3. That means if I have this chromosome, let's consider this is chromosome 9. Sorry for. Okay. Now say in this region is p-arm 1, 1. And say this is q-arm 1, 2. This is q-arm 1, 3. Okay. So from this region to this region, it was cut and it was flipped over and inserted. Okay, so it means inversion in chromosome 9 involving the p-arm and q-arm. So is the centromere involved? Yes, the centromere is involved because both p and q were involved. That means the centromere was involved in it. Okay, and such inversions are called as pericentric inversion around the centromere, pericentric. Paracentric would be something like this, involving just one arm. It does not involve the centromere. It's on either side of the centromere. Translocation. What is translocation? Let's say this green chromosome is chromosome 4 and this orange is chromosome 20. Now, a portion of chromosome 20 is broken and a portion of chromosome 4 is broken. This chromosome 4 is being attached to the chromosome 20 and this 20 is being attached to here at chromosome 4. So there is a translocation between the Q arms of chromosome 4 and 20. Okay. So what is this chromosome karyotype of? What is the problem in this karyotype? If you see, we have two chromosomes of all the autosomes, but one sex chromosome is missing. Of course, it can be a Y missing or X missing, but it's a X0. That means total how many chromosomes are there now? 44 and 1 X. That means we have total 45 chromosomes, comma, X and a 0. Okay, 45 X0 it is. Now, write the chromosomal and the karyotype as how many total chromosomes are there? 47. Okay, so we'll write 47 XX in bracket, we'll write trisomy 13 or whatever the trisomy is. If you look at this image, this karyotype, what is the problem here? 1 to 22, everything is in 2 copies, no problem. X is also double. Okay, good. So we have 46XX, is it? No, we have an extra Y there. That means there is a extra sex chromosome. Okay, so we have 3 sex chromosomes here, 2 X's and 1 Y, XXY. This is a 47 XXY. Okay, this is a 47 XXY, which is nothing but a Klinefelter syndrome. What is this? Orange has been swiped to gray, gray has been swiped to the orange. What is this? It's a translocation. Okay. Thank you.